In this video, I want to talk about two different globular proteins, and they are myoglobin, which is abbreviated MB, and hemoglobin. You may have heard of these two proteins before, or at least heard the names before. And we're going to talk about them and sort of their similarities, their differences, and what's important to know about them and how to understand what's going on with them. So we're going to first talk about and focus on myoglobin. So myoglobin, again abbreviated as MB. The first thing you might notice about myoglobin is that this myo portion, if you recall from before, not that I've mentioned before, just maybe of your own knowledge, that myo means muscle. You may have heard of myofibers, which are muscle cells, or myocardium, which describes the muscle of the heart, things like that. So the root myo means muscle, and this glob portion here implies that it's a globular protein. So first and foremost, myoglobin is a globular protein, and that's something we should keep in mind. And it's a globular protein that exists in muscle cells, and it is actually a water-soluble protein. Okay, so one thing that's very, very important to know about myoglobin, after I mention this specific thing here, it binds oxygen. Okay, so its ligand is oxygen. A ligand is just something that can bind something else, as far as the way we'll talk about it here. So O2, or oxygen, an oxygen molecule, can attach to myoglobin. So now that leads me into this idea of the function of myoglobin, because this is very, very important to understand. It's very key to understand this. The function of myoglobin essentially is to bind and release oxygen to the muscle cells. Bind and release oxygen to muscle cells. Okay, that is its function, and that is very, very important to keep in mind, and you'll see why in a moment. Okay, so like I mentioned, O2 is the ligand. Now, another bit that I think that you should know is that its structure is simply one polypeptide chain, which means one polypeptide chain, more specific, and within this one polypeptide chain, there are actually 8 alpha. Actually, maybe no, I'll put it like there. This one polypeptide chain has 8 alpha helices, which we've mentioned in a previous video. So specifically, this one polypeptide chain means that myoglobin has the highest level of protein structure that it has is tertiary structure. It does not have quaternary structure, right? Because it only has one polypeptide chain. Okay, that's an important bit to keep in mind. So this myoglobin, yes, has a single heme group. One heme group. What is a heme group? So this heme group is a prosthetic group. Oops. Which essentially, if I've mentioned before, it's actually a tightly bound non-amino acid component of a protein. There's actually what's it called? An iron ion, right? In this heme group. Okay. One more thing I wanted to mention is that there are no beta conformations in myoglobin. Sorry, I should have mentioned that earlier. Anyway, since it's a water-soluble protein, right? Since it's a water-soluble protein, the what's it called? The polar side chains are on the surface of the protein, right? And the hydrophobic side chains are in the interior of the protein. One last thing I want to mention about the structure of myoglobin is that it has 2 histidine residues, right, which if you recall are polar side chains, two histidine residues in the interior of the protein. Okay, one of them anchors the heme group, while the other one is helps bind the oxygen. Okay, and also it also this one here, it also reduces the binding affinity of some toxic gases like carbon monoxide. So it reduces the binding affinity of carbon monoxide. Right, carbon monoxide is a toxic gas. Essentially, the way it kills you is that if your muscles are not getting oxygen, right, because the histidine is binding carbon monoxide, what ends up happening is that if your muscles don't get any oxygen, or if your tissues don't get any oxygen, for instance, like your diaphragm, right, which is a muscle, you can stop breathing, and if you stop breathing, you'll die. Anyway, that wasn't very pleasant. In any case, specifically this histidine residue that anchors the heme group is the distal histidine, and this one here is the proximal histidine. Little nitpicky details, but that's sort of the idea behind myoglobin. Let's talk about hemoglobin now. So let's draw hemoglobin's chart over here. Hemoglobin, abbreviated as HB, it's also called maternal hemoglobin. The hemoglobin that exists in adults is called maternal hemoglobin. In another video, we'll talk about fetal hemoglobin and sort of what, how that comes into play as far as globular proteins go. So hemoglobin is also a globular protein, right? And we know that because it's hemoglobin, right? Now, if you're familiar with maybe if you knew that myo meant muscle, maybe you know that hemo, right? Hemo means blood, or is the root for blood. Hemostasis, hemophilia, if you've heard those terms before, they all have to deal with blood. Anyway, so it's a globular protein, specifically a blood protein, and it is also water soluble. It exists in our blood, right? So it must, and our blood is very hydrophilic, a lot of water, a lot of it is water. And so this is also a water-soluble protein. So far they're pretty similar. In addition, its ligand is also oxygen. Okay, so far these two proteins are identical as the way we described them. Now let's get into the function and kind of differentiate the two. The function, again, this is what separates the two a great deal, and I think is very, very important to understand. The function of hemoglobin essentially is to take oxygen from the lungs to the tissues, right? And the tissues includes including muscles, right? Okay, so this is very, very important. Okay, now as far as its structure goes, I'll get back to the importance of the functions and in just a moment, actually. The structure of hemoglobin is a little bit different. It actually has 4 polypeptide chains. Okay, 4 polypeptide subunits, of which 2 of the subunits are alpha subunits that are actually 141 amino acids long, and 2 beta subunits which are 146 amino acids long. Now they're very, very similar in structure, but it's the way it's denoted here is alpha 2, beta 2. So those are the 4. There's 2 alpha units, 2 beta units. This doesn't mean alpha helices and beta pleated sheets. Keep that in mind. These are just the sort of designations for those each individual subunit. Now each one of those. Actually, before I move on, before I move on to even more details, these 4 polypeptide subunits, the fact that it has multiple polypeptide subunits means that it has the highest level of structure is quaternary structure, right? Because it has more than just one polypeptide chain that makes up the overall structure of the protein. Okay, so that means it has quaternary structure. Okay. Now, what these 4 polypeptide subunits can do is that since there are 4 polypeptide units, each one can bind an oxygen. So this means that it can bind 4 oxygen molecules. Each one of these subunits, right, actually has a heme group. Oops, not each subunit. Each subunit has a heme group. Okay. So now, there are a few fun facts that you should be familiar with as far as hemoglobin goes. Okay. So hemoglobin's little fun facts are it displays this thing called cooperativity. Cooperativity. Am I spell this correctly? Cooperativity. Honestly, I can't spell. Anyway, it displays cooperativity. Spelling is I mean, important. I will agree, but I just want to explain this to you. Anyway, this whole cooperativity. What does this mean? Well, what I'm trying to get at here is that the whole idea behind cooperativity is that once it sort of goes back to this idea that it has 4 subunits. Cooperativity, because this hemoglobin can bind up to 4 molecules, once one subunit binds an oxygen, the affinity for oxygen at the other subunits increases. Okay, so let me mention that. So once one oxygen is bound at one subunit, the affinity for oxygen at the other subunits increases. It increases. So what that means, another way to say this, is that hemoglobin binds oxygen cooperatively. So to kind of give you a visual of this, imagine hemoglobin looking like this. If we have, just as a very, very rudimentary drawing, but imagine hemoglobin as this globular protein, and I'm splitting it up into 4 subunits. If we have an oxygen here, and it binds this subunit, right? The affinity for oxygen at these 3 locations will increase. These locations here, these other 3 subunits, will want to bind oxygen more than they would have had there not been this oxygen already bound to this subunit. So this whole idea here is sort of based on a concept called allosteric regulation, right? And we'll talk about that more when we're talking about enzymes. But allosteric regulation, just to kind of mention the sort of idea behind it, is that the binding of one ligand, right, or anything that can bind a particular protein, the binding at one subunit has an impact on the affinity at the other subunits for that ligand. Okay, so actually the term allosteric means other site. So other site or other location. So this cooperativity is an important concept about hemoglobin. Now let's talk about the 2 of them as far as which one binds oxygen better, right? So some of you may already know the answer to that question, but I'm actually going to just blurt it out now. Of these 2, myoglobin has the higher affinity for oxygen. Make it in purple. Has higher affinity for oxygen, right, than hemoglobin. Okay, so why? Why is that? Okay, well, if we think about it just in the terms of the definition, imagine this. You breathe in oxygen, right? That oxygen needs to go to your tissues. Let's just say in this case we're talking about your muscles, right? You breathe in oxygen, that oxygen goes to your lungs. Hemoglobin binds the oxygen in your lungs and takes it to your tissues, in this case your muscles. Once it gets there, it needs to drop the oxygen off. What's it going to drop the oxygen off to? Myoglobin. Well, that means that myoglobin has to have the higher affinity for oxygen if it has to grab the oxygen from the hemoglobin. If hemoglobin had a higher affinity for oxygen than myoglobin did, then hemoglobin wouldn't give up the oxygen to myoglobin. Okay, so let's write that down. So it has higher affinity for oxygen because myoglobin needs to grab the oxygen from hemoglobin. It wouldn't be able to do that if it had a lower affinity, right? So because myoglobin binds oxygen better than hemoglobin, it is able to actually steal the oxygen from hemoglobin. Okay. So now let's draw this sort of saturation curve, as they call it. Okay. Let's draw this out. Let's see what we got here. Okay. So now I'm going to do my best to explain this curve. So here on the y-axis we have this thing called percent saturation. Percent saturation. And on the x-axis we have the pressure of oxygen measured in torr. Okay, so what we can imagine here is I'm going to draw the lines or the sort of curves for myoglobin and hemoglobin respectively, and sort of kind of explain this as I go. So let's just let's do myoglobin first. Myoglobin saturation curve looks like this. Okay, it is a hyperbolic sort of graph. This little bend here is kind of me being terrible at drawing, but imagine this line is a little bit more straight and the slope is a little bit more, just that straight. So this is myoglobin here. Okay, hemoglobin looks a little bit different. Hemoglobin looks something like this. Okay, something like this. Okay, so what am I trying to get at here? Let me label these axes or with numbers here. So 0% saturation, let's just say 50% saturation is up here, and 100% saturation is somewhere up here. Okay, the PO2 is the pressure of oxygen. So basically, the higher the PO2 means that the more oxygen there is in the environment. Percent saturation describes how much of that protein is bound by that ligand. What percent? Let me actually note that here. This is the percent of the protein bound by a ligand. In this case, O2. Okay. So here, if you at this point there's 50% of myoglobin saturated or bound to O2. Here, 50% of hemoglobin is saturated. So what am I trying to get at here? I'm trying to say that if we have, let's say, a PO2 of 1, right? 1. That's a very low pressure. Whereas, let's say, a pressure of like 25 is over here. Okay. Now notice what we have here. At 1 torr, we have this percentage, right, of myoglobin bound, whereas at 1 torr we have this much hemoglobin, right? Or excuse me, myoglobin, excuse me. So imagine it like this. Myoglobin becomes more saturated than hemoglobin at a given pressure of oxygen, right? In order to get myoglobin to be 25% saturated, we only need one torr of oxygen, whereas that 1 torr only saturates myoglobin maybe, say, I don't know, maybe like 1 or 2%. In order to get hemoglobin to get up to 25%, we need to add 25 torrs of oxygen. There needs to be more oxygen in the environment to saturate hemoglobin, right? But at 25 torr for myoglobin, the saturation level is near 100. Okay, so it doesn't take much to get myoglobin fully saturated, but it takes a little bit more to get hemoglobin saturated, which sort of implies that myoglobin, because it's left shifted, has a higher affinity for oxygen than hemoglobin does. Okay, so let me note one thing that I mentioned earlier is that this is a hyperbolic sort of curve. Oops. Hyperbolic curve, whereas this is a sigmoidal curve for hemoglobin. Sigmoidal just means S shape. Okay, so this sigmoidal curve is due to the cooperativity, right, of hemoglobin. So what that sort of is how I explain this is that at low concentrations of oxygen, we don't have a very high affinity for oxygen here, at least with hemoglobin. But as you add a little bit more and more, once an oxygen is bound, maybe the affinity shoots up, right? So this is cooperativity happening right here, sort of in this area here, right? Once there's a high enough, once one oxygen is bound, the affinity for oxygen just shoots up, and then we have this upward curve that's going at a crazy slope. Okay, so that is essentially it that I wanted to mention about these. I hope that video was helpful, and I'll see you next time when I talk about hemoglobin and fetal hemoglobin and how they're different.